Welcome all listeners to another FSC radio podcast episode. My name is Aedan and I'm here today alongside Santiago and Thimo. And today we're going back to the basics. We're going to be talking about the fundamentals, but not just any type. For centuries, people have expressed their desire to make autonomous beings, from Frankenstein in classic literature to Pinocchio, alongside the famed digesting duck automaton of Jacques de Vaucanson. You may likely think of some of today's creations of life as the advancements in artificial intelligence, but this lacks a lot of the core requirements of life. So today instead, we will be talking about simulating life with just a few molecules in a flask. And to help explain this to us, we have Sijbren and Otto here today. Welcome.
Thank you.
How are you doing today?
Very good. Thank you very much.
We're very happy to have you. Dr. Otto, could you begin by explaining the research you do and maybe what inspired you to start studying that?
What we're trying to build in the lab is a completely synthetic form of life, but minimalistic life. So as simple as it can possibly be. And what inspired is, well, it's a combination of something that I was always intrigued by, as well as just luck, serendipity. So I was always fascinated by evolution, and I probably would have become a paleontologist had I not thought that maybe job prospects in chemistry would be better. So I took a rational choice at the end. Nothing against paleontology, I still love it. And then kind of forgot about being able to ever do anything with evolution as a chemist, because that's not normally what happens when you're running a chemistry lab. Until we ran into self-replicating molecules by chance, and from there on, that was an opportunity that one could even get funding for, and from there on we just kept working on this and see how far we can build from simple self-replicating molecules to, and can we push this to become more and more lifelike, and eventually maybe a minimal form of life.
Wow, I was wondering, these self-replicating molecules, like what does it mean that molecules can self-replicate?
Well, it means that a molecule with other smaller, simpler molecules in its environment can use those simpler molecules to build more copies of itself.
On the topic, maybe before we get too into it, how would you define life as a concept?
Yeah, that's a very difficult question. It's interesting that you may not think of this as a difficult question, because if you look around you, you can distinguish living from non-living things. In most cases, anybody can. There are a few cases like viruses where it gets a little bit more difficult, or actually a lot more difficult if you think more deeply about it. But if you really try to define life, and many people have tried, it turns out to be remarkably hard, to the point that we still don't have a generally accepted definition of life. So I will also not give you one, because for that reason. But at the same time, you can now argue, okay, if you want to make a minimal form of life, you don't know what it is, what are you doing? So we did define a list of characteristics. We said, okay, our minimal form of life needs to at least have this and this and this and this. And we're trying to work to get this list complete. It's not so long. So replication, but also the ability to convert material that you find in your environment to more of yourself. So that's in a way already replication, but also metabolism is in there. And being separated from your environment, so a cell as a membrane. So somehow you need to be compartmentalized. That's a third criterion. And then if something that has those three characteristics is able to undergo Darwinian evolution, so it's number 4, then our list for now is then complete. And whether it's then alive or not, you can still have a debate. And I think that sort of debate will maybe also allow then eventually to get to a definition of life. Once you have something that you can really poke at and say, okay, this is what's missing here, then add it. Are we then there? Then I think we might home in eventually on a definition of life, although it may also be that it doesn't have a very sharp boundary. So it's a little bit like, I always like to compare it to the color, say color green. So when is something green? Right? Everybody knows what green is, but there's, when you start mixing in more blue or yellow at some point, So maybe an arbitrary boundary, and and there's probably a gray zone between living and non-living entities that we on Earth at the moment don't really have populated, other perhaps than maybe with viruses. But once you start making minimal forms of life, I think you get into this gray zone first.
Yeah. How did you first come upon these self-replicating molecules? Was it like a goal of a project, or was it just like you said, the serendipity?
Yeah, it was pure serendipity. So we were after something completely different. Actually, we wanted to make molecules that fold in a specific way, a little bit like proteins fold to get function. So we wanted to do something like this, and we had some ideas to what sort of molecule would be good for that, and we tried to make it and didn't quite want it to be made. So with a PhD student spent about 3/4 of a year trying, and then we decided, okay, let's make at least a simpler molecule which should have the same basic ingredients and do some experiments with that. And that then turned out to, I mean, it was not immediately obvious what it did, but it turned out after a while that we figured out it was self-replicating. And then we forgot about the folded molecules altogether and went for that.
Something a lot more exciting. That is very cool.
You said that from when you discovered this molecule, it took a year to really know what you're doing. Is it really that it takes so much to really understand, or is it that now with the new techniques it's easier to know what is going on?
Well, I don't remember. It took quite a year, but it at least took some time before we figured out the behavior. Well, it's largely because behavior, like self-replication, is not the first thing you think about when something weird happens, because there are not so many molecules that do this. And certainly not, I think this was the first time anybody ever found one that they didn't design to do that. And usually the designs were based on how DNA is replicated. Our molecules work differently, so that also made it harder to understand what they were actually doing. Yeah, so initially it was a funny sort of non-reproducible experiment in that it sometimes did one thing. It made one type of, well, what it turned out later to be a replicating molecule, and in other experiments it made another. But we never got a mixture of the two. It's either one or the other. And that thing, if you do a chemical experiment, it usually either gives always the same, or it gives a sort of distribution, and you get an average and some variation around it. And this was like bimodal. You either get one or the other, and that was weird. I liked it a lot. The PhD student at the time, less so, because she was taught for the right reasons that experiments should be reproducible. So she wanted to reproduce the experiment and find out what. So then she realized that what mattered was, so when we run experiments, they're done on a stir plate. So there's a magnet that rotates. You put little vials on there, and they have little magnets in them, and they are rotated because they're sitting on top of this big magnet that rotates. So she noticed that if you put a lot of vials, and that was the attempt to make it reproducible, right? You repeat the experiment many times. She noticed that where you put the vial on the stir plate mattered, and that it was something that never happens. But in this particular case, the vials that were in the center of the stir plate, where they're stirred better because the little magnets inside the vial are better pushed forward by the big magnet underneath. They always gave one product, and the guys at the in the periphery of the stir plate, they kind of those magnets are not so well rotated by the big one. They produced mostly another molecule, and then she realized it has something to do with stirring, which and even though you some are stirred better than others, and normally as long as you stir it's fine. The samples were also homogeneous, so there was nothing chemically normally that would make any difference, except that it did. And then you start to scratch your head, so what's going on here? If there is something that is sensitive to stirring, there must be large things in there, and our molecules are very small, so there couldn't be just molecules. There must be assemblies of molecules in there that are so large that now they become, that stirring somehow affects them, so you can break them probably by stirring. So this is how far we got then, and then everything quite quickly fell into place. But it was this one observation of something that was extremely not common, and most people, I would argue, would not have thought about where the thing was on the stir. Wouldn't even have considered it and analyzed the data in view of where the vials were placed. So this is something that helped a lot to get this resolved, and was not an easy, a common thing to do either.
This really is fascinating. When was this? When did this happen?
2009, thereabouts.
Wow.
Yeah.
And since then, the field, I suppose, has suffered like many changes throughout time. So
For our lab, yes. I think the outside world just keeps turning, and there's still very, very few people that try to do what we're now trying to do, is make a synthetic form of life. I think there's only one lab, other lab in the world that really makes this as a research target. So for the rest, there have been a lot of work on self-replicating molecules, but somehow that never was taken the next step. So it stayed at the level of self-replication.
why is it so common for like? Is it not a field of interest in the scientific world?
It's very fundamental, right? So if you ask the question, okay, what can you do with these? So let's suppose you succeed, then what? It's not immediately going to give a product that anybody can sell. So there's no short-term sort of economic benefit. I think on the long term it may be very different, but that's one. And I think, but maybe but at the same time there's always the fascination with life. So the origin of life question, which is a little bit related, right? So what we're looking at is, can we make life? And we don't care what it looks like, life that we have now or that may have existed 3.8 billion years ago, how life started on Earth. It's not really directing our science. There are a lot of other labs in the world that are going after the question, how did life emerge on Earth? And that of the sort that then led to life as we now have it. So this is, that's way more popular for reasons that, well, maybe have to do with the fact that it's somehow easier to start with that, because you can approach that question from many different angles. You can look at the geology of Earth 3.8 billion years ago. You can do the chemistry, and you can look at phylogenetic analysis. So you kind of run evolution backwards and see where that gets you. Whereas if you want to build a synthetic form of life, particularly the ability of copying is not an easy one to achieve. And the people that try that, it was hard for these systems to see how these systems could become more than just molecules that would copy themselves.
And did you look at like this sort of the chemical evolution already, or did these molecules change somewhat like evolution does?
Yeah, that would
billion years ago.
I want to call it. I think there the word Darwinian evolution is immediately. The moment you have autocatalytic, so self-replicating molecules. If they make copies when they replicate, then you can select from those mutants. Then I think you can call it also Darwinian evolution and chemical evolution. But it's not taking place with living systems yet. They're just not living
chemicals.
molecules.
Yeah.
But Darwinian evolution concept is not necessarily only restricted to living organism. You can you have evolutionary. I mean, language evolves by Darwinian principles. There are a lot of and people have made computer algorithms that evolve by Darwinian principles. So I think you could call this Darwinian evolution, and indeed, in a very a rudimentary way, it does this. So you can have replication with indeed mutations. It's actually easier to make replication with mutations than without, and this is a problem we need to address. And then selection, if you design your experiment cleverly, then you can also do that. So you can select for some molecules over others. The limitation is at the moment, that's why I call it rudimentary, is that the systems make only relatively minor mutations, and you have a small range of molecules that can be accessed through mutation, and then you usually make them all at some point. And then you're done, right? And then there's nothing more to explore. And that's where, in my view, the biggest challenge in the synthesis of life, but also the origin of life, is how do you get from this rudimentary evolution of a very small set, where you explore a very small little landscape of possibilities, to open-ended evolution, where you can keep making new discoveries, new inventions that then open up further evolutionary space for yet new inventions, and this keeps going on. Perpetually, and that is what life does. And I think that may be the hardest part of life, if you want that to be, if your definition of life is requiring open-ended evolution, then I think that's probably the hardest part to make happen.
Yeah, because I'm assuming that if you put any molecule in there, it will likely not form. It will not have these characteristics of self-replication.
True. Yeah, I think that's also in a way helpful because it allows the replicating systems to use the molecules that they're not already part of some other. So I think that's not so bad. The problem is more how do you expand space in a way that it's not immediately all explored. So maybe a little bit difficult what I say now, but you want evolution to have many options, but at any point in time, not explore them all, right? This is what evolution does all the time. So you have life that you have certain organisms and they can vary a little bit. So you can have mutations that make them change a little bit, but not too much at one step. If it were too much, the mutations were too many, the organism would no longer function. So there's this balance of mutation rate versus well, you need to keep the mutation rate in check, basically. If it's too much, then nothing is functional anymore. And if you leave it to chemistry, the chemistry gives you too many mutations too fast. So you need to somehow restrict what chemistry inherently wants to do to reduce somehow the mutation rate, and that is not so easy. There's no clear. I mean, evolution solved this problem. Now we have a tremendously accurate way of copying information in our DNA, but it uses machinery for that has evolved. So it took a long time of evolution to get there, and this leads to a paradox. If you have and the machinery is itself very complex, so you needed a lot of information to build that machinery. So this leads to a paradox. So how do you get to the machinery that allows you to copy accurately as long as you don't have the ability to copy accurately? And yeah, there are different theories that people have developed. This is mostly done in the theoretical realm of how that could have happened in the original life and how, and we use those sort of theories now also to see if they can help us fix this problem.
And are you finding any success?
No, it's still early days. We know what to do. We haven't. So the trick is, so now when it's not to put all the information in one molecule, because if the copying of that molecule goes wrong at any one bit of your large amount of information, then you lose function of the whole thing immediately. So it's better to chop your information up in little pieces that then individually all replicate. And then they all make mistakes as well, but there is still a fair probability you have at least of every piece, you have one or a few functional copies after the copying process, and that's better than putting it all in one. So you want instead of instead of one replicating molecule, you want many that are different, but then you want selection to occur on the collective, on the community, not on the individuals. Otherwise, you would still not have solved any problem. So then that means all these individuals, they're all different. They need to cooperate so that the selection can take place on the community level, at the community level, and not at individual level. So that's what we're trying to do now. Can we make communities of coexisting replicators? And that's we can do a community. We can make 2 or 3, maybe 4, and can we then evolve those communities? And that's where we're at now to see whether that's possible and where that gets us.
Do you think, given enough time or I guess research on this, was it catalytic, you said, for the technical term for them?
So the self-replicating molecule is autocatalytic, so it catalyzes its own formation. So it basically speeds up the reaction that makes more of itself. That's what autocatalysis means.
Autocatalytic.
But now you mentioned catalysis. Catalysis is the molecules are not only autocatalytic; they're also able to catalyze, so make additional molecules, including the ones that they need to grow from. So that is like metabolism does. So it takes molecules you eat an apple, but your metabolism converts the molecules in the apple into molecules that you need to keep functioning. So we have that, but not with the same level of sophistication and complexity as metabolism in current life, but at least at a rudimentary level, this is what's happening.
Do you think if given enough time or research into this, these sort of rudimentary autocatalytic molecules could maybe not exist completely self-sustaining, or are they already self-sustaining? If you just left them kind of in a beaker to sit, would they be able to quote-unquote survive?
They are pretty stable, so they don't immediately fall apart. That's helpful, although they cannot be too stable because life also requires everything to be turned over at a reasonable rate. Your question is I guess more one of autonomy. So if you put them in a beaker, well, they need to be provided with an energy source and some resources, some food, if you like. Otherwise, nothing will happen. But that applies to all of life, right?
Yeah,
There's nothing without. If the sun goes out, then we're also gone with it not too long. So that there is, but then the level of autonomy is then, I guess, behind this question. So how good are they at doing this without our help? Well, for the moment, they're miserable. So we really need to provide them with very specific molecules because they cannot make those. They can maybe convert to something that they can use, but it needs to be pretty close already to what they require. So if we feed them glucose or an apple, it's not going to know what to do with it. So it cannot live off that. So if we know anything, any of our molecules, we set them loose and outside our lab, they have zero chance of replication, let alone survival. I mean, they're not alive yet, but so the autonomy is something that also is a bit of a definition question. When are you fully autonomous? And people say, okay, in life, what it does, it runs all the processes that need to be run autonomously. It still needs resources from the outside world, like food, like energy, but once you give it that, then that's enough. So that's the point where I guess we'd like to go to as well, but then I think the foods, again, are not going to be apples.
Don't you think that for some people it could be a bit scary to make life?
Yeah, I can imagine that, and we also we started to work together with Daphne Brandenburg of this university, who does research into ethics. The ethical questions surrounding this, while we're still well ahead of having anything that one needs to get worried about, so that we can chart what are the benefits, what are the risks, and what's the balance of those, and can we proceed? Now, for now, I think we can, but can we proceed also when we get more advanced? So this is an exercise we're doing right now, and we hope to have that develop faster than our work in the lab, and we make sure it does.
And you wouldn't have an issue with at some point stopping research at all, and then as long as you don't have if you have a clear answer to the ethical concerns, will you? Yeah, and if you don't have that, will you stop at some point your research?
We want to have at every step clear that step is not going to cause also further down the road unleash something that you don't want. And then, I mean, if there's a risk that is unacceptably large and not outweighed by the benefits of it, then you should stop, and we will stop.
Do these molecules in these systems, like you said, like they're maybe sort of like little populations or little groups of them, do they behave like groups, or do they just behave like separate things? Do they interact with each other?
Well, for the moment, they're still not interacting. We still need to, our plans now is to see if we can make them help each other by, for instance, catalyzing reactions, producing each other's food molecules, or food, converting into the food that they have in their environment to molecules that they need, that the other guys need to grow from, and vice versa. So if you can get some community of that sort, then that their selection should act at group level. But you need to have some form of cooperation, and it may be cooperation by sharing a metabolism. That's the way we're currently trying to do to get it to work. It looks like the easiest for our molecules to achieve, but there may be other options as well. And I think this is where there's quite a lot of theory out there as to how you can make communities cooperate, and we'll see what's the easiest. So we have an idea right now, but we're still in the process of trying to develop that, make it work.
And as you said, time again this is at the end of the day a bit of luck in the process.
There is indeed always an element of luck in this. I think you also, once you make a few of these lucky discoveries, and then you think back, okay, it was luck, but it was also, it happened in this system and it didn't happen in the lab next door. What was the difference, right? So what was in this system that made it possible for these self-replicating molecules to emerge? And then you realize that, hey, yeah, we put the right ingredients. We didn't know we did, but we did. So, and once you've gone through that experience a few times, and we did in the past also with other things, then you start to realize that actually, in order to favor luck, it helps to run experiments in a way that you maybe don't over-design them. You just put the ingredients there. You want something to happen, think about what ingredients are necessary. Don't think it through maybe necessarily all the way. I mean, you do as well, of course, but it may not be necessary. And it may even be better to not design it through. You're thinking through is one thing, but if you then make already the molecules very specific so that they can only do this, then I think you limit the probability, the chances of a chance discovery. So by just putting ingredients in an experiment, say, okay, what does it take to replicate? It turns out what it takes to replicate is exactly what it takes to fold, except that the interactions play out a little bit differently. So the ingredients we put there, we put for another reason, but they were there. So if we now want ingredients of like cooperation, you need catalysis. Maybe we shouldn't overdo the design too much and not specify this molecule to catalyze this reaction, that one is that, but rather allow the system the building blocks, or give it the building blocks with which catalysis can emerge once it finds the right way of putting those building blocks together. So at that level of design, I think we still rely on chance, but we also favor it a bit.
Some minimal building blocks would be there.
Yeah. You don't. I think that's just the lesson we've learned on several occasions the hard way, that you overthink a problem, you over-design the molecules, you spend a lot of time making them, and then it turns out the simpler ones were better or other. Yeah, that this behavior could have been achieved way simpler and in a better way. And indeed, this is also how it went with catalysis, for instance. We wanted catalysis because it's such an important part. I mean, metabolism revolves around catalysis. Well, if we eat something, we need catalysts in our body, the enzymes, to convert what we take into the molecules that we actually need. So catalysis is important. So we initially thought we have to design that in, and it turned out we just had to look better than what we already had, because it did not require any additional design to also get catalytic activity. That's also where it helps to be in a very nice environment here in Groningen. So Clemens Meyer, one of the colleagues here in the department, actually looked at our molecules and said, "Hey, you should have tried this reaction because it looks like this. Your molecules look very much like others in the literature that catalyze these reactions." So we did, and it worked way better than the things we tried to design.
Yeah.
Sometimes the simplest answer is the best.
Yeah.
Life lessons.
You said earlier that there might not be direct applications for this research, but for long-term applications there might be. What are some long-term applications you might see from this research?
Yeah, I always find it a very difficult question to answer because we're so far away at this point from those. But I'd like to draw a parallel. So I think we're now at a stage where chemistry was quite a while ago, before where people still felt organic molecules were molecules produced by living organisms. So that's where the term organic chemistry came from. And then it was with the synthesis of a very stupid, simple, deemed organic molecule, urea, which It's barely now even considered an organic molecule, but that opened a whole field that made people realize, hey, you can make organic molecules yourself. You don't need living organisms to produce them for you. And that then unleashed the whole, made a lot possible in chemistry. So one of the first things that people started to make were dye molecules, but also the whole pharmaceutical industry is now based on molecules that we make. We don't rely on living organisms to produce those molecules. So the idea that those molecules can be made by chemists, not necessarily only by living organisms in like cells, or that's generated a lot of possibilities that at that point, after the synthesis of urea, it was completely impossible to predict. So polymer industry, a lot of things happened after that. And I think we're now, if indeed it becomes possible to make a synthetic form of life, it will be like urea. It will be completely uninteresting and useless. And we have way more sophisticated, better and useful chemical living things at the moment already. So why bother, you could argue, except that I'm also convinced that it will do something similar to what the discovery of urea unleashed. It will allow suddenly possibilities that we could not imagine were possible. And now that's a long sort of attempt not to answer your question. So now I'll try to address it more directly. So if you can build a living organism, you can build it to do exactly what you want. And that could be clean up something you don't want in the environment, or produce something that you do desperately need. And you can now do it unencumbered by what evolution supplied all current life with. So you can tweak current life also to do some of these things, but only to a limited degree, because it still needs to do all its normal things to remain alive. And that means you cannot stray too far away from that. And I think if you build something from scratch, it could be much more purposeful and efficient at one specific task. That's my hope. But again, that needs to be proven, and that needs way more time and research before we're there.
Very difficult to imagine those stuff, because it's just so far in the future that it's like, yeah, I get it.
Regarding this topic of creating a synthetic life, in one of my in my ethics course in my chemistry bachelor, we had to write an essay about right-handed and left-handed bacteria. So for those who are not aware, evolution at some point decided that life was going to be right-handed, which is just left-handed and right-handed are exactly similar. The only difference is that they're symmetric through a mirror axis, which makes them function the same way, but they don't interact with each other the same way. So one of the issues that this essay we had to write about proposed was that left-handed and right-handed bacteria may interact differently, and we already know that from organic molecules in pharmaceuticals. But I'm asking to you, do you see this as, because this paper talked very, very big things about left-handed bacteria, how an unleash of left-handed bacteria could turn the world upside down. And if that is the case, then maybe we shouldn't discover these things. So regarding this topic, do you really think it's as dangerous as it seems, or do you think people are overreacting over this?
Well, I think the moment you make a bacterium that is exactly as evolved as a current bacteria, except that it's a mirror image, then it's pretty then you really have a problem with the competition between the two. Is not necessarily won by the ones that we like it to win, namely our handedness, our mirror image. So they're competitive, and I think the other side is the benefit of those types of bacteria. Why would you want to make them? What could it do? I think the answer to that is there are a few things that might be useful for, except that the risks are so high that maybe they don't outweigh the benefit. And I think that was also the conclusion of the study that was done by a large set of people that are working in the area of making synthetic forms of life based on DNA, RNA, proteins. But just to couple it back to what we're doing, we're not using DNA, RNA, or proteins. Whatever we make is so primitive that even if and no matter what handedness it got with mirror image, it would never be able to compete with current life, because the 3.8 billion years of evolution are not easily overtaken by something that we now produce in the lab. Even if it does become possible to achieve open-ended evolution with it, I think it lags behind so far that normal life, and that's maybe also why we don't see life forms on our planet that are not derived from a life form that already exists, right? In principle, well, at some point, something sprung up out of nothing. No life, there was no life on Earth, presumably, in the beginning, and then life emerged, and that was a spontaneous process. We don't see it happen anymore, and I think Darwin already had a good explanation for why, because anything that would spring up would be very primitive and would have no chance in the rat race against the highly evolved species that we really have on our planet. But then the mirror life is different there, because it's you basically you use exactly the same building plan, but just a mirror image, and that's going, if you succeed, it's hard. But if you succeed to do that, then I can see that be a problem.
As well, on the note of DNA, earlier you might have mentioned, or forgive me if I misunderstood, is there an application, maybe even if just theoretical, for building blocks like these to be used as some form of gene editing or CRISPR? In my mind, they're similar, but I have a very limited understanding of chemistry. Could this be somehow used in a way similar to that, or?
No, I think the crosstalk between whatever we have in our test tubes at the moment and current life is extremely minimal. So we use some of the same building blocks, but not even we don't have DNA or RNA-looking molecules, and we cannot definitely not edit DNA or RNA of a living entity with our molecules. There's no way of doing that, and I think the current tools we have for that are for good reason based on biomolecules and evolved biomolecules, because those are the molecules that through evolution have acquired the ability to do things like this. So we don't have in our systems, definitely not. And I think even if you were to try and make something that does what for instance, the CRISPR system does synthetically, it's going to be extremely hard.
Yeah, that does make sense. To be fair,
you were talking about how you're very far from replicating the evolution of life on Earth because we're so far ahead, 3.8 billion years ago it all started. But do you think your research is maybe closer to evolution of on other planets that could happen?
Yeah, it's an interesting question. I'm not sure. I think we know too little about other planets and what the actual sort of organic chemistry is that goes on them. To be able to tell, it would be an extreme coincidence, I would argue, if we were. But the universe is large. There are many other planets. We know next to nothing about them at the level of detail we need to answer that question. That I prefer also to not answer it.
You still mention organic life. Do you think a lot of science fiction books in the 20th century talked about the silicon-based life possibility? Do you think it's still a possibility that it could happen that way, life?
Yeah, I think the thermodynamics favors organic chemistry over silicon chemistry, in that the silicon oxide sand basically is a very stable form of silicon. And in order for life to run, it needs to continuously interconvert all its components. So it's life builds itself, but it gets deconstructed again, and it gets rebuilt from more or less the same, at least the same atoms to an extent, even the same similar molecules. If you have a sink for your atoms that are too stable, so it start to become sand or rock, it's very hard to extract those atoms again. So that makes me a little bit skeptical. On the other hand, if it means you need more energy, there are planets that are closer to the sun where the conditions are different. So maybe there, who knows? It's very hard to say. What I do find, the piece of information, again, that's based on Earth circumstances. So again, how generalizable this is, if you start to look for life elsewhere, it is questionable. But I was amazed to learn how few minerals there are. So if you look beyond organic chemistry, you think the periodic table, so you have all the different elements, not only carbon, nitrogen, oxygen, is large, right? And that must mean there should be way more possibilities to make molecules, to make chemistry happen with that part of the periodic table. Why only use carbon, nitrogen, and oxygen, and a few others? But then it turns out, if you look at the minerals that on this planet, there are a few thousand, and most of them are even derived, wouldn't even have been there if there hadn't been life. So the number of minerals, if you just leave a planet to do its thing, at least if you look at Earth, and if Earth is representative there, is extremely limited. And that brings back this point of what I referred to earlier. If you want evolution to work, it needs a lot of space to evolve into. And if the options are relatively few, as it seems the number of minerals is certainly relatively few, the number of organic molecules is extremely large. You can never make all the organic molecules that are possible, not even if you had all the matter in the universe. So large. So that, I think, is something that helps if you want to evolve, that you have this space that you can evolve into. And whether a non-organic chemistry offers a similar space, maybe not. But again, I think that's never been rigorously researched, so maybe I'm wrong.
There is another question that I wanted to ask. You've talked that it's pretty difficult to predict what the outcome of putting a few molecules in a batch is, but is it possible? Is it something that you're looking into with the advancements of artificial intelligence to maybe compute the molecules?
Yeah, the artificial intelligence is great if you have great data. So it's not like I can now I can go talk to my artificial intelligence colleagues here in Groningen and ask, hey, how should I fix this? I know the answer. I say, okay, first come back with the data. First go measure and then come back with a data set of at least 10,000 data points, and then we'll talk. Now there are also now tools that can do with a little bit less data, but still you need a lot of data before this gets you anywhere. And we are very in this field, there is a scarcity of data. And but then you could argue, okay, then you can set up a automated experimentation and just collect that data. That will be an option. We haven't dismissed it at this point. But we haven't prioritized it either, because I think at the end, evolution, once we can get it to work, can create stuff and maybe better at developing new things than artificial intelligence is at this point. I think at this point you need the data, and then it kind of can interpolate. It can do things like the data showed, but can it do new things? I think that's where evolution is still better, where you can actually, if you challenge a system that's capable of evolving with a problem, that if it doesn't solve it, it dies, then it either dies or it solves it. And if it solves it, it has developed something new. And yeah, that's where we are most interested in. And maybe that's also, I'm a bit biased because my fascination of evolution comes back here, but I'd like to see that happen and see how you can get from something very simple to something more sophisticated through evolution.
On that note, are you sort of trying to influence mutations to come about in a certain way, or for like trying to find combinations of molecules, for example, that may give you better long-term stability or better ability to replicate? Or are there certain factors that you're trying to propagate more in these molecules, or are you kind of just trying to see how it will, quote unquote, naturally come about?
No, there are a few things we'd like the system to develop. One is mentioned a few times now, catalytic ability. So can you, by evolution, increase the ability to catalyze? The other is accuracy of replication. Can you become more accurate, make less errors? So if we want to influence mutation, then yes, but then fewer mutants, please. because at the moment they're making too many errors when they're copying. So if we don't somehow restrain that, then so we need to restrain the number of options. Otherwise, it just basically melts away in all the different things that it does at the same time. And yeah, that and then what I already mentioned is building these communities. So can we do this in some way? So how should we mix the building blocks such that they become different coexisting replicators rather than one that uses all the building blocks? Questions like this are really pertinent right now.
So this is kind of what your group is researching right now. Just make those parts more efficient.
Yeah, and we still need to. So we have replication, we have a compartment and metabolism, but we're still working very hard on integrating all three. So I think we're almost to the point that we can write the first paper where that actually happens. So that's still hard. Also, that's the groundwork, but that still occupies a lot of our time. And then the next steps are in the direction of evolution of those systems that have a compartment, catalyzing reactions, and are also replicating, and then make communities of those. So that's that's pretty much. Different people are working on different aspects of that big picture.
So then, soon enough, once you have this, you could, according to your definition, call it that you've made synthetic life.
Well, I hoped I was careful in my definition to say that, well, you need evolution, but when are you satisfied with evolution? I think to me, that's where I would draw a line somewhere and say rudimentary evolution, so you can make 3 mutants and you can go from one to the second to the third and back to the first. I mean, that's not going to, I would not call that alive, because this is not capable of becoming anything. It will always be that. It will never be anything else. So the ability to evolve properly requires this open-endedness. So you need to convince yourself that after seeing a few inventions that this can continue forever. And that's where I would draw the line. I mean, if I see that, I say, okay, now we have created something that we've seen we started it at one place, and then it evolved to invented new things 2 or 3 times. And I can see that this now, with the new invention it made, at each step or at several of these steps has opened up yet more opportunities for the system. And this could go on forever, which is then a prediction, which you would ideally want to verify, but that would then literally take forever. So that will never happen. So you need at some point to convince yourself, okay, this is good enough that I'm now confident enough that this is essentially open-ended, and then I would proclaim victory. But we may not get there, because I think this is not an easy problem to crack, and we may find that our system has just not the ability to become sufficiently complex. The way it's currently set up, we would the amount of information that can be stored and copied is still very limited, and it would never get there. So we have some ideas as to how to make the information content bigger, and in principle also unlimited. It can maybe a bit more specific. Certainly at the moment we store our information in a ring, and the ring can only be of a certain size, and that means that's the physical limit on how much information you can then put. What nature does, and what we should also do, is not put information in a ring, but in a linear thing that can be as long as we want. So the ring we know we cannot make as big as we want, but the linear thing we could in principle make as long as we want. If we can succeed in transforming the information, or transferring somehow or making the step from rings to these linear constructs, that's necessary. Whether we will be able to do it, we'll need to find out.
Why are those rings not be able to grow bigger? Why are they limited to a certain size then?
Yeah, that's a good question. I think they become too. It doesn't fit anymore. The architecture is such that it's kind of, the information is sitting on the ring and is protruding radially outward. But the moment you start to make the ring bigger and bigger and bigger, then these radially outward protruding things no longer have space. They become squeezed together to a point that just in each other's way. So that's why I think that architecture is fundamentally not good for getting very large.
Because I was asking because obviously life also has a ring size information structures like bacteria and stuff.
Yeah.
So I was
cyclic
like,
RNA, cyclic.
So yeah, it does exist. But yeah,
I was wondering how often do you look back to biology to take inspiration for your designs?
I mean, evolution, of course, is something you take inspiration from big time. We like to borrow bits and pieces of biology. So our molecules have amino acids. Amino acids are the building blocks of proteins, but we don't build proteins from these amino acids. We use them but the way they interact are similar to how they interact in the protein. So we can use a lot of what people know from protein chemistry and use that in our design. So that's what we do. We're happy to borrow bits and pieces. What we don't want to do is borrow anything from biology that is already highly evolved. So an entire protein or a DNA sequence or something like this. So we'd like to borrow the chemical building blocks, and they, you can argue, also are selected by biology probably from many options because they were good for certain things. I'm happy to borrow that and take that further. But then we like to run the system and let the system do what they want to do with that.
When do you predict if it ever happens that you may create what you were mentioning earlier, this infinite evolution, like for an infinite amount of time? Would it take decades? Would it take
time scale? I mean, if I look back on where we are now and how long it took us. So it was 2008, 2009 when by chance we found these molecules. So then we worked on replication for about 10 years and then found out they're catalytic. And now that we know they're catalytic, I think we're another 5 years down the line and we were able to make the compartments also. And in the meantime, we've done the early evolutionary experiments with not yet in the compartments, but with separate replicating molecules. It's been picking up pace, I would say, but whether that necessarily means we're very close to getting to anything that is open-ended evolution, we would need at least another 5 years. That would be a very optimistic scenario. The pessimistic one is we'll never get there because our system is inherently unsuitable, and that wouldn't then be a very easy way to start again because there are not so many systems. But we may have learned something then, and we may have. But then it would be beyond my my retirement age. It's hard to predict. It's too many uncertainties.
As well, on the note of the enclosing membranes, is that formed through? I mean, the only example for me that comes to mind is the phospholipid bilayer, I think, in like animal cells. Is it something somewhat similar to that uses kind of polarity, or what mechanism?
We don't. We tried, played around a bit with that, but it was very difficult to get those sorts of compartments. So, but instead we use a different type of compartmentalization, which the current cells also still have, and they've become very topical. They're called liquid organelles. So they're basically assemblies of usually large molecules, and usually it's because they interact in a funny way. They like to stick together, but they're a bit frustrated. But they can't. Normally, when molecules like to stick together, they phase separate, so they become a separate phase at the bottom of your vial that doesn't talk to the rest of the world anymore. These liquid organelles are also molecules that like to interact with one another, but they're a bit frustrated. They can't do it all the way, so they don't completely phase separate, so they become little droplets. There's still a lot of water in them because there's also sites in these molecules that still want to interact, but they couldn't do it with the other guys. So they're little droplets, still a lot of water, and a lot of frustrated interacting sites that they want to bind to other molecules with. So if you can give them those other molecules, they tend to go into these droplets. So they're then making compartments that have the tendency to, so if you can call a droplet then a compartment, that have a tendency to enrich material that is there free in the surrounding solution, and that happens spontaneously. So we don't need to find clever ways of putting things inside. It just goes there spontaneously and is there at much higher concentration than. So where we got to so far was with our self-replicating molecules, we're able to catalyze reactions that form the molecules that make those droplets. And then those replicating molecules also go inside. That part we kind of expected. And then you have your compartmentalized self-replicating molecule. So in a way, that's not too dissimilar from what you also still see in cells. So the inside of this, if you look inside what's in this bilayer membrane with your phospholipids, what's inside is also very many of these types of liquid organelles, little droplets of material can be found inside the cell even today. So it's not like we invented something new there. This was known, and actually one of the earliest theories of the origin of life also is by Oparin, and then he called it coacervates. They're the same thing: liquid organelles, coacervates, membraneless organelles. And he saw a great potential of these things in the origin of life scenarios. So the idea is pretty old to use that as a type of compartment. It's probably the first proper origin of life theory actually revolved around this. So it's nice to see it revive now. So not only by us, there are also many other people working on these types of constructs and see what they do. And yeah,
These coacervates is what you think in your research group is the best way to mimic life.
Yeah, for us it works for the moment very well. And the advantage is, a membrane. Maybe at some point also necessary, we'll have to see. But the coacervates don't have a membrane, so things do go in and out relatively easily. Things concentrate up inside is what you want. Neither of these things happen, both of these things are very difficult with membranes. Making things go through a membrane is hard. You need, usually in the cell, there's a protein that does this. Now, when you make something simple, you don't have these types of sophisticated proteins. So that's one, and concentrating things up inside also needs an active pumping mechanism, because inside is water, outside is water. Why would anything want to be more concentrated inside than outside? So you need to pump it in if you want it to up concentrate there. That needs an energy source. You need coupling to this energy source. Again, you need a very sophisticated something that, again, at the beginning you don't have. So then it's better to use something which does what you need it to do and doesn't need help, just does it autonomously.
It really sounds amazing that this can happen just in a flask. Is fascinating.
Yeah, it is. It absolutely is. It's again, when you give the system the freedom. It's at the end we know it's possible, right? Life uses the same tricks. So the question is, how do you make that happen then in the test tube as well? And I think it's by specifying some things, but by not specifying some others, and giving it also the freedom to do what it wants to do.
What, at this point, is the scale of these molecules? Do you have an example to compare it to, or on the scale of, is it microns or nanometers?
So the replicating molecules, they are a few nanometers in diameter. So this is extremely small. They can be up to a few micrometers in length. So there are rings that are stacking on top of it. In the screen. There are rings that are replicating or stacked on top of each other, and the stack of rings can get very, very, very long, very large. But they then sit inside these droplets, and you can see under the microscope. So they're micrometer size, and now it gets also. One of the challenges of chemistry is you work with most of the molecules that people work with are so small you cannot see them under a microscope. The really big ones you can see under an electron microscope, but most of the molecules you also cannot see under an electron microscope. So there's no direct visual way of looking at what they're doing. But now we're building these cell-like entities that are large enough that you can, at least at the level of the cell, see what happens. Although at the level of the molecules inside, we're still the things that are a few nanometers thin, you cannot see on the light microscope either. So it's they're still quite a challenge to study what these molecules do at a molecular level. At the cell level, we can see, but at a molecular level, we can't.
What techniques do you use to learn about these molecules within the?
Yeah, whatever we need. So we're when we were still working with the individual molecules, a lot was based on chromatography with mass spectrometry. So chromatography is is separating molecules based on their nature. I don't know if any of you have ever done this experiment where you put a dot of a black marker pen on a filter paper and let it then run in an alcohol solution or something, and then you see that all the different colors that the black color was made of appear as different spots on your filter paper. That's the principle of chromatography. Now you can do this with filter paper, but we have somewhat more expensive ways of doing it that are a little bit better at pulling things apart and then telling what they are. We do by mass spectrometry. So once you have one of these dots in your filter paper, you can put them in a mass spectrometer, and it tells you how heavy that molecule is, what's its molecular weight. And from that, you can then work out what probably the molecular structure is. In most cases, the molecular weight is enough for us to know what we're dealing with. So that was for the molecules, but then when you go to the assemblies of molecules, then you get to a larger size range, and on the chromatography the assemblies fall apart into the individual molecules, so you cannot study the assemblies with your filter paper approach. So then you need to go to like electron microscopy. So we have a very good electron microscopy facility here with a very good electron microscopist. So Mark Stewart helps us a lot with that, and that helped us learn about that. And then also like high speed AFM, so this is a technical term, but that's kind of another microscopy technique where instead of shooting electrons at it, you kind of feel over the sample with a very, very sharp needle. So there's a group led by Wouter Roos here in Groningen that's one of the top end instruments, and knows how to handle that. So together with him, we looked at how, and we were able for the first time to actually see, and with this technique, how these replicators grow. So with electron microscopy, you get like an image, but you cannot capture the dynamics. You cannot record a movie. Here now you could actually really see how things grow. That was really nice. And now that we're looking at these cell-like entities, then just optical microscopy, but confocal microscopy, so you use techniques that you get more detail in your image, is now used a lot to figure out what goes on there.
Crazy techniques that you guys use.
Yeah, I think this is also the field of chemistry and science at large has now developed to a point that when I was a PhD student, if you want sophisticated mass spectrometry, you needed to go talk to somebody who did this for a living. Now we just have an analytical department that has the mass spectrometers in there, and anybody in the group can use them hands-on, right? So there's electron microscopy is almost got to that point as well, and a lot of people in the group, not everybody now is trained, but a few people are trained, they can do this themselves. So the techniques have advanced to a point that I think we can now do this, but 50 years ago, that wouldn't have been possible yet.
So it really is only right now where this field could have been developed.
Yeah, I think it's fair to say that we would have really we probably would have got stuck 50 years ago at some point, and we wouldn't have been where we are now.
Yeah, you probably wouldn't know what was going on.
Yeah, I think it's fair to say.
As a closing question, maybe for you, what is or has been or will be, do you think, the most exciting thing about this experiment for you?
I think it's the finding out how these systems can just develop themselves, realizing that it's indeed possible that something like life just can emerge. It's not necessarily a very probable thing, but that if the right circumstances are there and the right molecules, I mean, the laws of nature are such that you can get molecules to do these bizarre thing that we now call making something that is alive. I think it's you can now see a path there that was completely obscure when we started.
Very, very exciting.
Thank you so much for being here.
My pleasure.
It's amazing conversation, this. So yeah, really, thank you so much.
Definitely. I think it puts a lot of things into a different perspective in terms of kind of Earth and everything.
Thank you.
Thank you for having us.
See you next week.
Bye.